Hello.
Good morning.
Morning. So, how are you this morning?
Good, just pre-coffee.
Oh, yeah. Yep. So, yeah, we didn't have our Friday meeting yesterday. Had a conflict, and we had DevoWorm this week, which was good. Talking a lot about the graph neural network stuff, and talking about connectivity. Mahul presented on a paper that he was interested in, so we had a lot of discussions about that. So. Right. So as you know, GSOC is completed. The normal time period for GSOC is completed, and Sarah Bostawala was the student in the Orel lab. We have two students in DevoWorm who aren't finished with their projects. They got a 3-week extension, so we'll be talking about their projects later. But then we also had Shubham Soni, who was working on like a reinforcement learning model of open source sustainability. That wasn't funded by GSOC, that wasn't accepted by GSOC, but he nevertheless worked on it and pushed the materials to our repo. So there's Sarosh. Hello. Lukas.
Hello.
Hi, how are you?
Fine. How about you guys?
All right. This is actually Sarah's last blog post. This is her GSOC 24 report, from concepts to completion. So this is really kind of going over the final framework, LLAMOSC, that's large language models for open source sustainability. It's actually based on the Llama open source large language models. Talking about how those were used to generate agents and using agent-based modeling to generate contributors that address tasks that were also generated through a large language model. So this kind of goes through a lot of the work that she did, kind of from this diagram that she presented in early meetings here to some of the implementation components. So like the environment variables, agents, agent variables, which were the internal states. Then there's the framework capabilities. So she integrated auto code rover into the framework, which is something that generates code. So part of it was to generate tasks, but it had to also generate code. And so the code was in response to the issues that were created. So this was just a lot of generative, kind of basically generating an open source community, maybe a small open source community, and then seeing how well it worked given certain conditions. So that's why she was integrating AutoCode Rover, integrating this automated pull request lifecycle with a contributor agent and a maintainer agent. So the contributor would generate some solution to an issue. The maintainer would check the code to make sure it was consistent with the issue. And then we had these multi-agent decision-making algorithms. So there were two algorithms that kind of governed how the contributors operated. One was the authoritarian algorithm, or the benevolent dictator model, where the maintainer manages and allocates tasks, and then the contributors are rated and assigned based on what the maintainer says. And the decentralized algorithm, which is the meritocratic model, had distributed decision-making amongst contributors, not maintainers, and the contributors bid on tasks based on their suitability and experience. So once the agent that represented contributors had a profile, they could then bid on tasks. They had a certain amount of experience, and then that was done. The maintainer, of course, is also automated, and in the case of the authoritarian regime, would actually do the allocation of tasks. So that would impact how sustainable or what the open source community would look like. Then there were metrics and trends visualization. So she had a number of things here. Where she had this pop up where when you run the simulation, you set a number of parameters, So you have the number of contributors, number of maintainers, the number of issues. Those are the initial conditions of the simulation. Then you set the decision-making algorithm, and you select an issues path, so you can select certain issues. You can hand code the issues if you'd like, or you can generate them, and then put them in this file that you can import into the simulation. And then this is for the calculator project that Sarah created for this. This is as a demo of this, and then starting the simulation. So it runs a simulation, and you get these results based on the initial inputs. There are also some metrics that are used in the simulation. We can plot those out. So all of this is present in our open source meetings from this year. So we had open source meetings from May to this month. So we had our last meeting last week. We weren't able to have one this week. So thank you to everyone who participated in our open source meetings. They were weekly affairs that were very useful for students working in open source projects, people like Morgan who have experience in open source, and Jesse, of course, working on his project management issues, and then myself, who was leading the whole thing. And I talked about a number of topics in open source. So I think that was very useful. This fall, we're going to do something related to modeling and data science at that time. So it's going to be a different thing. So I feel like, that was a successful season of open source, and looking forward to doing it next year. The other thing I wanted to highlight was this was the Shubham Shoni's repository. So this is Shubham, and he's been contributing a lot to the open source sustainability, or a lot to the GSOC repository in this directory, open source sustainability using RL. So if you go up to the GSOC repository, we have this is, of course, Sarah's work here, and this is Shubham's work. If you go into this directory, you'll see that he has the PI file set up and the README, which actually doesn't have anything in it right now, but I guess he's going to add those in a little bit later. So this is, so he's issued a number of pull requests this week, which is why I wanted to highlight in the meeting. So there were some updated files here, and so this is good work as well. I think he still needs to do a little bit more, but I'm not sure. It's fine. There he is. But yeah, congratulations to Shubham for also doing a great job on working on the reinforcement learning model that he was working on this summer. So it looks like Shubham's here, and so is Jesse. So Shubham, hello.
Hey everyone.
Hi.
Am I audible?
Yeah, you're audible.
Yeah. Just give me a second.
Okay.
I have some network issues here. But regarding the updates, I don't have any visual updates. I have pushed a lot of code into the repository. So that is mostly the previous work, and I have added some RL logic into it. So what I was trying earlier was I will use Pettings to create a custom environment, but implementing that would require a lot of it would require a lot of maintaining variables. That was not really worth it. But what I did was I reached out to the developers at MISA, and they have actually completed the reinforcement learning project that they were supposed to do last year. So I reached out to them, and they have done some work in implementing reinforcement learning into some MISA models of their own using RL library. There's another RL reinforcement learning library called RL library. So I've been trying to implement the reinforcement learning part with the help of whatever work they have done, using it as a reference. And probably by this weekend, or at most by next Saturday, I'll be done with the entire thing, and then I can have I can display two different models, one with reinforcement learning and one without it, so we can compare the results between both of them by next week mostly.
All right. And so, yeah, you were talking about that in the open source meeting. So the non-RL model is just doing things at
Yeah.
random, right?
Yeah, non-RL model is doing things at random, and when we implement RL, it'll have a reward system, so it'll take its actions based on the policy and the rewards.
All right. That sounds good. And we
So
did.
maybe
Oh, go ahead.
at most a week, and then
Yeah, a week. That'd be fine.
could be a week.
Yeah. So we had talked about also Sarah building a lot of these automation tools. Have you looked at any of those for this project, or?
Not yet. I was just hoping I'll finish this RL part first, and if anything, if you guys have any suggestions for the automation, so we can do that as well.
Well, that sounds good. Thank you, Shubham.
Thank you so much.
Yeah.
Also, I have pushed the entire code, whatever the updates are there until now. So if you guys want to review it once, you can just check it.
Yeah, I saw that. Yeah, there's you made a number of pull requests this last week, so that's good.
Yeah.
Thank you.
Thank you so much.
Yeah, thank you. So I don't know if Jesse or Lukas wanted to give an update. Does Lukas want to give an update?
Yeah, I can give an update.
Okay.
So, yeah, basically, what I did was that I started working on the coding part of the Jupyter notebook. I developed a pipeline, and I started doing. I started running the blast searches using the pipeline. So as of right now, I've ran all of the blast searches, which is like a blast protein blast, and I've ran it for around 25 proteins now. So I use the NCBI accession IDs from the paper, and also around 5 more proteins that are involved in C. elegans developmental, like C. elegans development and stuff like that. So what I did as of now is that I finished the primary data collection. So right now, if you look at the GitHub page, I have the Jupyter notebook there and all of the JSON files that we needed for primary data. So yeah, and I'm also looking to use the primary data so like basically the OpenWorm studentship had 2 goals. The first goal was to develop the Jupyter notebook and pipeline and collect the primary data, and the second goal was to use the primary data to achieve a secondary goal, which we're still thinking about what we can do with the primary data. So yeah, so as of now, this primary goal is achieved. However, we're still thinking about what we have to do with these JSON files and what we can do with the blast search results. So that's my update on the OpenWorm project.
All right.
Project.
Yeah, we talked, and I know we've been talking about what to do with them, and I'm like kind of cagey about that. But I'm really trying to figure out what to do. I mean, I'm still kind of working that through in my head how we want to implement that. But so I guess the next step would be to kind of do a presentation on this project, like just kind of going through the notebook and the JSON files, and then I know that that Porg wanted to see, what had been done and everything, and so we'll try to schedule that in where you can present on that. And I'd like to see it first just by myself, so I can give you feedback. And then I can either share the video with Porg or we can do another presentation with Porg. But I wanted to get like a good, presentation, I don't know if you want to do demo or slides or what, but, to sort of summarize everything and sort of lay it out there for people, for what's there. And I think that's valuable because, it's kind of hard to hard to say. First of all, it's hard to say what we can use it for if we don't know exactly all the, functionality of the tools and so forth. But also because it does summarize everything that was done. And, if you look at the GitHub repo, you might not, actually get what's going on. So, I would like to see that. And
Oh
yeah.
yeah. What I can do, like, I was thinking about this actually. What I was initially thinking was to like when the project is all done, I can like just record a video just by myself or with you together to go over everything that I've done, and then this I can also present it in a meeting too. But like we can save this video. We can either post it on the YouTube channel and post it on the Slack channel too for others to access it. I can also put it on the GitHub page as well, just in case people want to see how it works and stuff like that. But yeah, I can do both. I can record a video to go over everything, and also I can present what I've done live too, just in case they wanted to see how it works and stuff like that.
Yeah, that would be good. Let me know. You could just kind of do a video on your own, and you could send it to me, and I'd look it over and see, make sure it's got everything in it. So or we could do this as part of DevoWorm even, or this meeting. Just let me know. We'll do it in the next few weeks. I don't want to make you do it like right now. I mean, I'm sure you're starting your semester. So yeah, this is good. And I was looking over the GitHub repository, and of course, I don't know if are the Jupyter notebooks up there yet, or
yeah, it is. So like I haven't worked a lot with GitHub pages, but yeah, I think it's like I was thinking about like I don't know how this happens, but the the Jupyter notebook is in between the zip files, so people cannot really see it at first. But it's called OW Studentship with the IPYNB. It's called owstudentship.ipynb, which is the thing for Jupyter notebooks.
Yeah.
So yeah, that's on the GitHub right now with the zip files. But yeah.
Yeah. So this is the fork of Lukas's OpenWorm studentship project on DevoWorm. So we have this zip file which contains JSON files for each protein, and then this is the notebook. I don't know if you can render the notebook.
Oh, this one apparently it's not updated for some reason. Like I have a different version, which is the more updated one. Maybe I have to. This is the one. I don't know why it's like the forking had a problem or something. I don't know.
Oh, I just need to update the fork, but I haven't done that yet. But
okay.
Yeah. But this is So this is just, these are the proteins here in these zip files. So the zip files, I think, have like the JSON files, maybe like 2 JSON files per protein or something. And then this is the notebook. And so this is good. And so, we can go over, I guess you can go over maybe, what's in the repository, go to the notebook, and then, kind of maybe walk through the notebook, and then maybe walk through a single zip file to show the JSON format and like how that is structured, and also, kind of what that gives us in the notebook. So like it's pulling in a JSON file. There's this API that's, maybe even talk about a little bit about the API that you're, the source of the data that you're getting. So it's coming from somewhere. It's coming from a database, and you have, the database is assembled from different people's primary data that they've collected and uploaded there, and they have it in this format, and you're taking it down and putting it in JSON format, and then bringing it into the notebook. And so that, I think, would give people a good appreciation for what was involved in the project.
Yeah, great.
All right, thank you. So I don't know, Lukas. You said you also wanted to present on a paper today.
Yeah, I'm ready to present that if you want. I actually uploaded the PDF file of the paper on the Slack channel. It's in the general channel. Okay. Yeah. So can you guys see the shared screen now?
Yes.
Yeah. Okay. So yeah, the paper that I chose to present is called the Comparative Genomics and Functional Study of Lipid Metabolic Genes in C. elegans. So basically, what this paper mainly does is that it goes over like they've collected 471 lipid metabolic genes in C. elegans, and what they do they're basically trying to achieve two goals by publishing this study. The first goal is to develop a network of metabolic pathways using the 471 lipid genes, and they're the first to do it. So basically, these authors used the 471 lipid metabolic genes and assigned each one of them to a metabolic pathway, and they actually integrated each of the 16 pathways into a network, which they go over later. But yeah, so basically the first goal was to develop this network, and the second goal, which actually is on a deeper level, is to basically their second goal is basically to suggest that C. elegans could be used as a model to study lipid metabolic diseases or even other diseases, human diseases. So yeah, so basically the first page goes over the abstract background, and it's basically a summary. However, what I wanted to what I wanted to mention is a couple of terms that not a lot of people might know about. Maybe they know, but I just want to go over them. The first one is ortholog. So basically, a orthologous gene or orthologous genes are genes that have that basically are from the same common ancestor, but they've been separated due to a speciation event. And there's also another. So basically, they're homologous, but they've been separated due to a speciation event. There's also something called paralog, which is not mentioned in this paper, but basically a paralog or paralogous gene is a gene that, or genes that have the same common ancestor, but they've been separated due to a gene duplication event, which is also mentioned in a lot of genomics papers. However, this paper mainly focused on orthologs. And there's another thing that has been mentioned in the first page, which is really important. So basically, in the results section on the first page, they talk about RNAi or RNA mediated interference or RNAi. Basically, RNAi. Well, basically, to look at RNAi, we also need to mention something called gene silencing. So basically, the idea of gene silencing is that a lot of researchers try to either turn off a gene expression or decrease the amount of gene expression to understand the phenotype or a biological function of a certain gene. So basically, there are two types of gene silencing. There's something called gene knockout, which basically is gene knockouts completely turn off a gene expression. So there are different technologies used for gene knockouts, like talons or CRISPR, and they basically turn off the gene expression completely to understand the phenotype. But in this case, they use RNAi, which uses the concept of gene knockdowns. Gene knockdowns basically decrease the amount of gene expression. So they do not completely turn off the gene expression, but they decrease the amount of gene expression. And RNAi basically is a technology used to do gene knockdowns. And basically, based on my understanding, they used RNAi because, first of all, it's I talked to a couple of researchers about it, and apparently, RNAi is a little bit cheaper to use than CRISPR or TALENT. So that's one of the reasons they used the gene knockdown method. The other reason behind them using RNAi technology was that they it's, as they said, they're about more than 85% of the primers were available, so they didn't need to start developing their own library for gene silencing. They just like the primers for different for the like a significant amount of lipid genes was already available. So they basically used the already developed libraries to start doing gene silencing. So that's another reason why they used the RNAi technology, which I thought is kind of important to mention because a lot of people don't know about it. So yeah, basically the first page basically goes over the background. There's a lot of stuff related to biochemistry mentioned in the paper too, like fatty acids, what they are, lipids, what they are and what they do, and why they're important. And basically, yeah, to give you a summary, this page, the first page, basically goes over the entire paper, just summarizes what they're doing, and yeah, so that's the first paper. The first page of the paper, and the next page, page 2, basically tells you a lot about how these researchers obtained the 471 lipid genes. So as you can see on figure 1, they obtained 168 lipid genes from a database called KEG. I think it's pronounced KEG, if I'm not mistaken. Yeah. This database, basically, this database is also another database for gathering like genes. But the unique thing that KEG does is that when you search for genes on KEG, it gives you it basically gives you the pathways that genes are involved in. So that's a useful thing for these authors because at the end of the day, they're trying to develop metabolic pathways. So they might have needed the genes. They might have needed this additional data. So that's another reason why they use KEG. So they use 168 genes from the KEG database. They used 147 human ortholog genes. Basically, I think the main database for gathering the 147 human orthologs was, I think it's also another tool named Biomart. So Biomart is a tool used. It's used in a lot of for a lot of things. It has a lot of useful tools. But if you go on the Biomart website, one thing that they mostly use it for is finding human orthologs. So yeah, basically the majority of these human orthologs were gathered from the Biomart tool, and also 156 of these 471 genes were gathered from the literature. Well, it says literature, however, yeah, they didn't gather all 156 from the literature and the published papers. So as you can see on the bottom of the in the description of the figure, it says that 156 genes manually picked from wormbase.org and published literature. So yeah, basically, they picked the 156 genes from wormbase.org, which I think, which is also related, which like is an organization that has done a lot of research on C. elegans too. I think their main goal is to develop and contribute to the C. elegans community. So that's one thing, and also yeah. So Figure One basically mentions how they got the 471 lipid metabolic genes, and it also yeah. So it also mentions basically on the here. It basically mentions that the 471 lipid genes in C. elegans were not only involved in distinct lipid metabolic processes, but also play multiple roles in response to oxidative stress, aging, and longevity, as well as other biological processes that are also kind of important when researchers are studying human diseases. So yeah, and at the last paragraph basically goes over basically looks at the 471 lipid genes and mentions the big lipid families that they got this lipid genes from. So basically the main lipid family was well, one of the main ones was lipase. The other one was phospholipase, and there's also a family called P450 or CYP, which is also another huge lipid family. So yeah, basically, page 2 also goes over the results and mainly the lipid genes. Yeah. Page page 3 you can look at the table 1, which basically they're summarizing the lipid metabolic gene families in C. elegans, and the other column like there's the genes, the enzymes related to them, and also the number in database. So basically they found the number of genes, and they look at how many genes there are in total and how many of them have been mapped in pathways. That's also kind of important for them because they were trying to develop that metabolic pathway. So they're trying to but basically summarize how many have been found and how many have been mapped in pathways. The paper also mentions that some of them are well, basically the paper mentions that some genes were found, like basically out of all the 471 lipid genes, some of them could be specified to a certain metabolic pathway, but some of them couldn't because, for example, the CYP gene from the cytochrome P450 family, it's the paper basically mentions that these genes from this family do not have a specific biological function for them. So like there is not a biological function that has been assigned to some of those genes, so they couldn't really specify or assign those genes to a metabolic pathway. Yeah, and the rest of this page basically goes over the lipid metabolic pathways. One of the more important things that they mentioned in this page was the fact that some of the genes, such as like some genes had to be, well, basically they mentioned the exceptions in the assignment of genes to pathways. So like they mentioned the CYP33E2 gene that basically initially they found initially the KIG database gave them a different result than what the actual function of the gene was. So they had to reassign the gene to a different pathway, and they go over other genes too and mention, well, some of them they mentioned that they had to reassign because of confusions and different database results, and some of them they couldn't even assign to the lipid metabolic pathway. So basically, that's the third page, and the fourth page, which I mean, I'm a little bit disappointed because I guess they could have kind of. Well, it's a great figure. It's a great network. They've assigned all of these metabolic pathways, but it's really hard to look at them. You know, it's I had to even I tried to. Yeah, you have to zoom a lot. Basically, if you've printed this paper, you can't read it. You can't. So I guess what they could have done was that they could have added the figure to an appendix or did something better in terms of representation of the network, because it's really hard to read. But yeah, that's the figure 2 is basically the network of these 16 lipid metabolic pathways. So if you look at here, it mentions that the blue ones, basically, like if you look at let's say linoleic acid metabolism, here, if you look at this pathway, basically the blue ones, like it's really hard to read the gene. I think it's like W07A8.2 or something like that. These genes are the genes that the blue ones are the ones that have been gathered from the KIG database. The orange ones are human orthologs, and the red ones are the ones from the literature. And the green dash line, which is really interesting, is basically representing the link to different pathways. So there might be a certain product in a metabolism pathway that is a substrate for another, metabolic pathways. So what they basically did is that they connected the substrate and the product from different metabolic pathways using the green dash line. So yeah, basically that's figure 2. And the other thing that this this page goes over is the biomart, which is I think it's right there. The biomark. So basically, they mentioned that they use biomark to find orthologs in human, mouse, rat, and Drosophila. So basically, they use the 471 genes to find 581 human orthologs, 585 mouse orthologs, 563 rat orthologs, and 428 Drosophila orthologs. And they also, basically this this information is the starting point to look at the conservative genomics part of this, or conservation genomics part of the paper. So basically, if you look at this this paragraph, they actually mentioned that over 70% of the C. elegans lipid genes have orthologs in humans, and they mentioned that about 72.12% of human lipid genes are conserved in C. elegans, which is a relatively high relatively high percentage of conservation. Therefore, that paragraph also basically highlights the importance of studying C. elegans studying C. elegans when looking at lipid genes or lipid metabolic diseases because of this high conservation percentage. And this paragraph basically the conservation of C. elegans lipid metabolic genes section basically goes basically they look at a database called OMIM. I think OMIM is Online Mendelian Inheritance in Man database. Basically, it's a database used to look at disease genes, mainly human disease genes. So there was 19,994 MIM IDs. Basically, MIM IDs were the genes that they could find on the database, and they basically this section. If you look at this section, it's on page 5. Basically, they mentioned that out of all of those 19,000 or like close to 20,000 genes, they found 15,181 genes that could be considered human disease genes. And basically, they found out that out of all of those 15,000 genes, 1,264 of these may be considered metabolic disease genes. So basically, they found they used the OMIM database to find the amount of genes that could be considered metabolic disease genes. And they use keywords such as obesity, diabetes, metabolic disease, and other major obesity-related diseases. Basically, they use these keywords to find the 164 genes, and they one of the most important things that they found out was that out of all of those genes, about 346 orthologous genes are conserved in all 5 organisms. So it means that there were 346 genes that they found in this database, in this online database, and 346 of them were conserved in all 5 organisms in C. elegans, mouse, C. elegans, mouse, rat, Drosophila, and in humans. So that's also an important number because it also mentions that they, basically due to this high conservation, it mentions why they're using these. Basically, it's a number, but basically it's a high number of conserved genes, which later on is used in figure 3 to explain some conservation genetics results. And the most important part in this page, in my opinion, is this paragraph where they mention that in humans, 97 of the 581 lipid genes are overlapped with metabolic disease genes. That means that they were like, if you remember, the 581 lipid genes are the human orthologs that they found using the 471 lipid metabolic genes and lipid genes in C. elegans. So the 581 is basically the human orthologs. They found that 97 of those 581 are overlapped with metabolic disease genes. That means that out of all of those human orthologs, 97 of them could be human metabolic genes. And they say that similarly, 94 of the 471 lipid genes in C. elegans are orthologs of human metabolic genes. And they later on mentioned that crucially, all 94 of those C. elegans genes overlap with the 97 the 97 human metabolic disease genes, which also another basically they are proving the point that C. elegans are important models in studying human diseases because of this high overlap of a high overlap of genes and high conserved genes. And yeah, basically the other they go over the results and numbers, which I just summarize the most important information there. But the other thing to look at is figure 3. Figure 3 is basically using it's really interesting. They used a program called Venn to develop these Venn diagrams. At first, it looks kind of terrifying, but it's basically, if you look at like figure 3a, figure 3a is basically looking at basically human dash CEL is well, basically there are 5 like, yeah, it could be like 5 kind of circles, and it's a Venn diagram. So they look at commonalities. They look at sometimes like sometimes some of the genes are not conserved, like 44 of the human and CEL are just not conserved, so they don't have any commonalities. Similar to the human slash Drosophila, you can also look we can also see that the 44 of those ones also don't overlap with any of them. But the most important thing to look at is the 5,085 genes, which is an important number because it's basically represents the amount of genes that are conserved in all 5 organisms, which is a high number basically. This 5,085, if you look at it, you can see that it's common in all of the circles. Therefore, it represents the conserved genes in all 5 organisms. Basically, 3b also has figure 3b also has a similar concept, except that in this case they're looking at figure 3a looks at the lipid genes in general, but figure 3b looks at the metabolic disease genes. Figure 3c, d, and e are very straightforward. You can see the 97, like in figure 3c, you can see the 97 genes that are basically common in human metabolic diseases and human lipids. Similar 3D also represents the 94 genes. And figure 3E, again, basic Venn diagrams, but there are also, well, the 3A and 3B Venn diagrams are a little bit confusing, but C, D, and E are useful Venn diagrams in my opinion. And yeah, basically, the other section of this page looks at the RNAi or the gene silencing method that they use. As they mentioned, the out of all the 471 lipid metabolic genes, only a small number have been biochemically and functionally characterized. That's why they needed to use the gene silencing method to look at the biochemical details and functions of the other genes. So basically, the next page basically looks at how they did the gene silencing using RNAi and the results. Though the most important part in this page is table 3. Basically, table 3 is a summarization of these long paragraphs that are on page 6. So if you look at table 3, there's sequence name, there's gene name, and there's LDS. Well, basically LDS is lipid droplet size. And there's a column named other phenotypes, and the pathway and the key references. Basically, they looked at these genes. They used RNAi, and well, if I go, well, I can explain the lipid droplet size better here. So basically, if you look at figure 4 on page 7 of the paper, you can see that they've, well, first of all, they used a method called Nile Blue, I think. I'm not sure. I think it was Nile Blue, if I'm sorry, Nile Red. So basically, Nile Red is a dye used to well, it's mainly used for visual representation of different lipids, and it's basically a dye, but I think most people have heard about Nile Blue. Basically, Nile Blue is well, basically Nile Red is a substitute of Nile Blue. Basically, they boil Nile Blue with H2SO4 or sulfuric acid to get Nile Red, and then they use Nile Red for basically as a dye for lipid drop for basically the visual representation of lipids. So as you can see, there is a control here. I think I can zoom in on that. There's a control. They used a lipid droplet as a control, and then they use the RNAi on different genes, such as the SAMS1 gene, the LPIN1 gene, and they looked at that lipid droplet, and they reported the lipid droplet size on table 3. So basically, if you can look at like let's say SAMS1 gene, it had increased lipid droplet size, therefore it affects the growth rate, and so they could look at they could mention growth rate as a phenotype, and they also mentioned lean and sterile. The ATGL gene increased lipid droplet. Some of them, in an interesting part was that some of them had decreased lipid droplet size, such as LPIN1 gene. But the most important part, well, the most important phenotype and the most mentioned one was growth rate. If you look at these genes, you can see that growth rate was mentioned in most of the gene phenotypes. I think the paper later on mentions that out of the 21 genes that they looked at using Nile Red, 19 of them affected growth rate phenotype. So basically, regardless of lipid droplet increasing or decreasing, these genes had an effect on growth rate. Some had positive effect on growth rate, some had negative effect. So yeah, basically, that's a summarization of table 3, and then as we mentioned, figure 4 basically looks at inactivation of 10 lipid genes by RNAi. and looks at fat storage. They have a control there, so they can look at what happened to the lipid droplets in different genes. And yeah, this part is the discussion section. The discussion section mainly goes over the like basically is again a summary of what they've done. They basically I have gone through most of the discussion section there, but basically the other thing that I wanted to talk about is figure 5 on page 8, which I think a lot of people who are part of the C. elegans community have seen this type of pictures. It's basically that's the control one is an empty vector, and they use they basically use the control to look at the they basically look at the development of C. elegans and how those genes affect the development of C. elegans. And yeah, so basically this part of the paper also basically also mentions there are two important parts that I wanted to highlight. One of them is this part for they basically mentioned that C. elegans and humans share high conservation in terms of lipid genes and their roles in metabolic diseases, suggesting that C. elegans is likely a useful and insightful model for use not only in research on the fundamental biology of lipid metabolism, but also exploring the mechanisms of human metabolic diseases. So yeah, they basically again mention the importance of C. elegans as a model, and on the other paragraph, they mentioned that among the 471 lipid genes, nearly 40% genes were reported to affect phenotypes. As an example, they use fat content, Dauber, lifespan, brood size, etc. I also wanted to explain what Dauber is. So basically, if you look at the developmental stage in C. elegans, it starts basically there are two basically there are two versions of C. elegans life cycle. There's one version where you assume that C. elegans the organism is in a good environment. Basically, a good environment is described as having enough food. So basically, if C. elegans is in an environment where it has access to E. coli, which is its main food, they'll start as L1, go to L2, L3, L4, and then the adulthood stage. So basically, that's the C. elegans life cycle on an assumption that C. elegans is in good environment. However, if the C. elegans start in a bad environment, basically the bad environment would be the lack of food. Basically, they start as L1, but they go to a stage called L2D, and then a stage called Dauber. A Dauber stage is basically like a hibernation state, where C. elegans is in that state until they like they're put in a good environment. If they're put in a good environment, they go to the L4 stage and then the adulthood stage. However, if they're not, if their environment doesn't change, I think they stay in that Dauber stage. Also, another thing that I wanted to mention about the Dauber stage is that basically it can only happen before before L2. So before puberty, they can enter the Dauber stage. However, after puberty or after the L2 stage, if you change their environment from a good environment to a relatively harsh environment, they you basically might destroy them. But yeah, that's basically the Dauber stage, which they mentioned, which is also an important concept in C. elegans developmental stages. So yeah, that's page 8 of the paper, and page 9. Again, basically, there's a lot of info here, but basically, these paragraphs look at the genes, and they look at the OMIM database results. They've also mentioned the Nile red staining method, and basically that's all about lipids and what they've done. And as a conclusion, they conclude two things again: the fact that they've created a database of 471 C. elegans lipid genes. They found the human orthologs. They found that over 70% of C. elegans lipid genes have human orthologs. And they also mentioned that out of all of those genes, they used 21 lipid genes to study on more depth using the RNAi method. So, and the reason why, it's really interesting. The reason why they used 21, but basically, out of the 471, they had, I think, access to about 356 primers. So they basically tried using RNAi on 356 lipid genes, which is a little bit time consuming. But out of those 356, they found 21 lipid genes, and they studied it in more depth. The reason why they chose 21 was that those 21 were the main ones that had a significant effect on fat storage or development or reproduction. The rest of them either did not have a significant effect on the phenotype, so they couldn't study it, or it was basically, or the primer was basically not available in the library. Yeah, and the last part basically is the methods section. The methods section again goes through the construction of the database and how they found the 471 genes, and they also the next page also mentions the annotation of lipid metabolic genes, how they annotated it. They basically used a lot of important tools that I think a lot of people can also use, and it's really important. To go over all of the tools, one of the tools that they used is Venn, which is used for Venn diagrams. Another tool that they used is the Biomart, which they use for orthologous genes and finding orthologous genes. They use the OMIM database, which is also a great tool. They use KEGG database. Basically, I found that this paper also gives you a lot of insights on different tools and how to use them and what their importance are. So basically, they didn't go over it in depth, but they basically name all of the tools they used, and I think it's useful for other researchers as well. At the end, they go over the growth condition and RNAi. Basically, they mentioned that the standard protocols were used to maintain the C. elegans N2 strain at 20 degrees Celsius with E. coli, and they also go over the they basically go over the details of the protocols and the Nile red staining, methods. There's also a growth rate and sterility analysis where they give references to the growth rate analyses. You can look at the references section for that one. And they also mentioned that all images were captured using identical settings and exposure times. Ones which clearly laid fewer eggs were recorded as having a egg-laying defect, and those with no eggs were recorded as sterile. So yeah, that was basically basically the paper.
All right, that's great. Thanks for the presentation. I know that was a lot of stuff. Maybe people didn't really understand everything that was going on. I think Lukas kind of laid out a lot of the take-home messages in that paper. So they're doing like genetics. They're getting data out of a database to look at what they consider to be conserved. Then they do annotations so they know the function. They fit them into these metabolic pathways. Then they do these assays, which are the lipid metabolism phenotypes. They do the RNAi, which basically knocks down the expression of a gene, and then they make comparisons between C. elegans and humans. So the reason why we use C. elegans as a model organism for medicine is because it's easy to work with. You get a good generation time. You get to do things like look at sterility. You get to do things like look at lipid metabolism in a very simple way. You just take a microscopy image and you look at the droplet size, lipid droplet size, and you can assess from like the wild type or the control if it's larger or smaller. But as you mentioned, there are a lot of cases where you can't definitively say. And we have these defined mutants in C. elegans. So there's a whole database of defined mutants where people have taken genes and knocked them out, and then they can characterize the phenotype very easily. Well, not for all of them, but for most of them. And we know we can map that to the genome, so we know kind of when we knock out a certain gene. And there are a number of defined mutants for like metabolism, where you have if you knock out that gene, and this is through not RNAi, but a more complete knockout process, that you have this strong effect, the strong phenotypic effect. And so so you can use defined mutants, you can use RNAi, which is cheaper and allows you to do that in context, so you can look at different metabolic pathways. And that's all, very easily manipulable in the lab. So finding the connection between C. elegans biology, the manipulations that you can do in C. elegans biology, and then the human disease pathways and the human disease phenotypes is very useful. So, we have different medical models for C. elegans, like in aging, in metabolism, of course, in some other disorders. You'll see C. elegans used quite a bit in human medicine studies, and you'll wonder why we're doing this. It seems like it's a very distantly related animal, and the answer is just because we have these orthologs, we have these these highly conserved areas with respect to function that are not only conserved with humans, but with mouse, where we do a lot of other biomedical research and using mouse and rat as analogs for humans, because they also have very similar, they have, conserved genes, they have similar physiology. And so, we don't want to do these experiments on humans. We can't slice them up and do all these things, but we can do it with these other organisms. So very good, Lukas. Thank you for that presentation. I know that was something you presented in the journal club, and now you wanted to present it here. So I think it's a great overview of it.
Yeah, and if anyone has questions, I can also kind of answer to the best of my ability if you guys have any questions. But yeah, that was basically the paper.
So yeah, that's great. Now, I don't know if Jesse is available right now.
I don't think we had any real meetings this week in the conventional sense. I did a little Joe Pro related stuff at a broader level. No major updates, but a few things with the mental health working group. Jen's still working on a bunch of stuff there, which I appreciate. And I guess the biggest real news of this week is more like an administrative research coordination level. I met with my old department, or I guess the college within the university. And it was really good. I was actually really, really impressed by them and where they've gone. I haven't really been in touch the last year or two or so, and some things have changed and everything else, but people have kind of left and come and gone and all that. But the department itself and what they're doing for like experiential learning and outreach and creating opportunities for students is very promising. Like they've addressed actual concerns I had as a student and I had in the past. So kudos to them. Kudos to CEHC at Albany for stepping into some things and making happen. Basically, it was just really a catch up call, but like we have opportunity to do some internships kind of things. They even have like a particular opportunity where I don't know, a modification of opportunities where they realize a lot of things don't start at like August, September 1st. So they have like midweek, they have it so you can start certain things in the middle of like the semester, for example, to create more flexibility for organizations and students to start internships or learning, experiential learning opportunities and so on. And things like that, I think, are quite like, that's a huge barrier to reducing friction for many students, and I respect that. So, functionally, what does this mean for us? I'll be talking with them again soon, and not, different colleges or different departments and, well, I guess colleges and other things like that will have some different opportunities, but there's a lot we can do in terms of society ethics tech, some things in data science in general. I might do some data ethics things there, some those spaces. And then Bradley also mentioned about model builder as an internship. And I think we've talked about that. I think we even have a description somewhere in the past, but there's the college will have they look quite promising in terms of their ability to have some niche things that aren't just informatics student doing informatics internship or whatever, like very clear cut, like they are open to doing some like interdisciplinary or adjacent related things as well, which is promising. But then also understanding that there's opportunity to do it with the main campus center for other things that may be more like predominantly like psychology related or cognitive science related. We can do through the main campus like research center. But it was good to meet with these folks, and I'm happy to. I don't know if we want to talk any more about this now, but I think it would be good to talk about the model builder thing that Bradley specifically mentioned, and some other ideas and opportunities we have there in the future. But the summary is that it was I was legitimately impressed, and I think there'd be some nice opportunities ahead. So it was good.
Yeah, I know you mentioned that in the Slack, and I kind of thought, well, that would be a good opportunity to do the model builder as a piece, like an internship piece. Also, yeah, I think society ethics tech, that's been something that has garnered some interest in the New York Celebration of Women in Computing, so that's a natural or obvious thing to do. Data science stuff, that's what they do there as well. And you have some expertise in it now, and so yeah, we can do that. You'd have to kind of figure out how to, kind of what, I guess with data science or something like that, we'd have to figure out what people want to do specifically. It's not easy to kind of just say, here are some things, why don't we work on them?
Yeah.
If you whatever kind of you get a certain amount of expertise out of that, but if you had a specific project we could tie it to, then that makes it a lot easier. And we don't have although we have some stuff we could do in the group, I think it's better if we try to find something that we can put together, if they want to work on something in a platform, kind of work out a model, have some data available that they can mine or something, that would be good. Instead of just kind of saying, find your own thing and go for it. And, we'll spend half the term kind of figuring out what people want to do.
Yes, I agree with that very much. And I think I wrote this somewhere, but it's an important point. What's interesting, previously my work with the former version of these groups at Albany, we were very limited in what we could. We basically had to have one extremely generic posting. Mm-hmm. Now, with the way things are currently, we actually can do much more, especially for data science, society ethics tech, data science, ML, like the groups that I mentioned will have an opportunity to do much more individualized postings, and especially for what Bradley said for data science thing, I think that makes a lot of sense. But I think we could even do, there's a complex systems lab in the college. There's a few others that are particularly relevant to what we can do, to what we do in the lab as it is. And so I'm going to be investigating what those are, and either offering a partnership or some kind of a not partnership, but maybe some kind of a joint thing, or at least seeing what kind of research opportunities are already being offered and making ours, fit that mold a bit. But we'll have the opportunity to be a lot more specific, which I think will be nice to do. I suspect for the other main campus center, we'll have one. I think they'll be a little more generalized, but in this college, we'll have the chance to do more detailed things, which could be quite exciting. But yeah, I'll know more about that over the next week. So.
Okay, yeah, that would be great. I've been actually working on the cards that we mentioned, I think it was last week or the week before, and I've been updating those, and just looking at them, I was like, oh, these are really out of date. Yeah. So obviously I needed to update. But I've actually generated some for some sort of encapsulating not just projects, but kind of themes in the lab. So I've been working on that. I'm not ready to share it yet. I'll be working on it more maybe this weekend, but we'll go over those soon. And those will be, I don't know if that's necessarily the best kind of description, but I'm kind of working out some descriptions so that we can, if we don't want to use those cards, we can move that to some other medium and, kind of just give people a taste of different things that we do or we want to do in the
lab. I think one thing that I'm particularly interested in doing is consolidating, especially I think the book review that you've done, I think will be a great lead-in to the developmental brain vehicle project. I don't know, whatever we want to call that. All of that stuff, that's not even necessarily something we're going to pitch. Although we could somewhat yield it, but I think that's a coherent project thing that we can do. And I need to update Google some of our code stuff. We've had a lot of nice contributions and discussions and all this stuff happening for that. But like, I need to update that page. I've had to move things around in the Jerper website a lot, and that was sort of where the main archive was. But I'll that will be updated too. But I think that the Breakenberg vehicles is a really cool space between that and also these sort of some conversation with Avery, who is not often at these meetings, but we talk frequently, and there's a definite overlap between some of what Avery's trying to do, and essentially, like, I think there could even be like one of the things I wanted to do and never really fully did. I think I forgot what I called it before, but basically, like, I apologize because all the terms that come to my mind are terms from everywhere else and not in the lab, so they're going to sound really out of place. But almost like a center or a group or something focused on simulation. It's basically representational brains and phenotypes. Let me just call it that. Like, representational brains and phenotypes was the sort of banner that I originally came in underneath. It was about developmental brain vehicles. It was about even, what was it called, like contextual geometric structures and stuff like that? Is that old school? Those came up in a discussion this week kind of about some AV and some other topics too. It's like all of the stuff from that era that I think is, maybe it's Dev AI now or whatever it is, but like this sort of the metabrains, developmental brain vehicles, some things in the CGS category, like something there that's about simulating these things and like creating, like, what can we take away from, I don't want to say the uniqueness of this lab, and that we don't, we're not doing like wet lab stuff, but access to certain like computational simulations and things we can do that way, and like having something about that specifically, I think is something that I may try to focus on, but it requires some other things to develop more. But yeah, like all that stuff is on my mind.
Yes, let's please revisit the. I mean, I like the idea of having this overarching approach to the representational brains and phenotypes. That's still a thing, by the way. That's like the overarching theme, because we have a website for that, and that's where things get updated for all those things. So it's just like, you have this right now. You have this sort of large collection of work on modeling, agentive modeling and other cognitive science type things that we're doing, which always tie into that usually. And then, so that's all kind of in that area. Now, it's just kind of there as like a name on a bunch of things, but yeah, it would be nice if we could like stake out some sort of, saying why we put all these things together in the same pot.
Do you think this is kind of a side question, but I do, like, I know a lot of talk has been about, like, you said agentic modeling, agent-based models, some of this stuff. I feel like that's maybe a good term, but also like I think do you have comment or agree or disagree with that agentic modeling, or make like say like that would be more of a subterm rather than the whole thing? Like, I still think the best like overall banner is probably the original representational brains phenotypes thing, right? Yeah, okay.
But it's just, I'm just saying what's in there. Like,
exactly. Yeah. Okay. That's what I thought. I'm thinking about how to, like, all this like weird administrative discussing, like presenting stuff, which is always awkward and like inherently incomplete. So that can be a whole discussion to have later. It doesn't have to happen now, but I think that's there's stuff to do there, and I will try to be put that in the pipeline of stuff that's like happening adjacent to the internship, adjacent to reaching out here, adjacent to the stuff I'm doing with UCSD, which is a little bit they're like 3 or 4 or 5 weeks behind what we're doing here, so it's Yeah. Anything? I have like one other slight update, but
Okay,
anything else you want to say about all that stuff?
not right now.
My other slight update is I'm making slow progress on a number of things that come up, well, in my life, and one of them in the lab. Like Paola, if we recall, was like interested in this finding an organization to do some of that grant. Like I think it was her model cards idea, or
Right.
like something about the NSF grant for certain kind of learning or something. Um, I think I'm not I'm not uh, I'm not the best person to speak on where that is. I understand some of it. I think they're still looking for a grant, an organization basically to manage that. So between that and I think it's what is it called? The Future of Life Institute has the grant I was particularly interested in about like AI power or whatever concentration. I think I'm gonna try. I think I'm gonna. I don't know if the timing is gonna work out for stuff, but I have at least taken it a little bit seriously and started reaching out about who to do what. And I finally found the right people at UCSD who would tell me like their sponsored research office basically, and like their grant. I I will be getting some familiarity at least talking to that office and like having them show me the ropes about what is it like to get that kind of a grant from an external group and use it in this way as whatever. Which I would love to win the grant and have a wonderful experience with that. That might help this lab and or JoPro or other projects or whatever on varying ways. It wouldn't be gigantic, but it would be something, an experience. But I think the experience of working with the grant office will be beneficial to a lot of things I or we are trying to do later on too. So that is in motion. I don't.
Okay.
It's kind of been a bumpy ride because they're on breaks, but the deadline's ahead of time, and then U Albany is in a different way, and I'm trying to sort out all this other stuff. So things are in motion. It's exciting. It's weird, but that's the nature of it.
Well, that's great. Thank you, Jesse, for the update. So this is the Representational Brains and Phenotype website.
Woo!
Yeah,
It was
you can
back
see the
then where I used to see it.
representational-brains-phenotypes.weebly.com. It links from the Orthogonal Lab site. So if you go back to Orthogonal Lab, it's under NeuroAI. So we have people collaboration. You go to NeuroAI. We have this representational brains and phenotypes. We go to that. And this is an image I made from like a rainbow image of neurons and some agents on top of them. But the idea here is that we're kind of pooling all the work that we're doing on these computational models, embodiment, agentive models, even stuff that's bio-inspired, to kind of bring that together into one place. And so this is, we have a couple of categories here: cultural computational modeling, which actually is something we were doing. We were doing the contextual geometric structures, but we also I did a number of papers before that with Hrishikesh Kulkarni on some things in machine learning and kind of working on cultural things in machine learning. So there are a lot of variations to this. We have talks, papers, preprints, GSOC stuff, etc. So we have that. Then we have growth and form, which is kind of things having to do with this is more devorm related, but computational models of related to sort of organismal growth and form, but also with respect to incorporating those into embodied agents. This is kind of one of these things that's kind of very loose, and it's so the super performance paper presented at Beak AI is in here, but we have some other things as well. The critical period stuff kind of falls into this category. So this is this is all just kind of work. So we have like these categories, and each category is a work in progress. And so as we kind of go through that, the motions on writing papers coming up with ideas that kind of fit into these areas. So, there is no real synthesis here. It's just kind of all these different areas stuck together. Sometimes it feels like there is no organization, but they're related because you can relate them. It's not that they're fully synthesized and realized. So then there's the phasic IA, which is this phasic, which is physical intelligence, augmentation systems, and complexity, and intelligence augmentation. So this is an area that we haven't talked about a lot, but I have a map of it on the orthogonal lab. If you look at the carousel on the main website, there's a sort of a map of what that is and how those things relate to one another. So this is, some talks. This is from Neuromatch, talk that I did on human-assisted AI, and some other blog posts, some publications. So that's an area. And then finally, cybernetics and complexity, which is the work that we've done on the agents and agent-based models and embodied agents. So things like Gibsonian information, intelligence offloading, and developmental agents, metabrain models, developmental brain vehicles, embodied continual learning, and even some things on cybernetics in the embryo. And then, of course, the stuff on good regulator theorem as well kind of fits into this. So that kind of overlaps with what we're talking about with cybernetics. And so those all kind of fit together. A question is how do they fit together? And how do they, we've talked a little, in some of the papers, we've talked a little bit about something called developmental neurosimulation. But that's like one thing that's kind of unites maybe, say, the stuff on Breitenberg vehicles. But there's more synthesis to do there. Mm-hmm. And so, that's what we're striving towards. But that's like kind of one of the big areas of the lab. The way I have the website set up, I have like the orthogonal lab site, and then we have these projects. Some of them are administered by Jesse, some by me, and we have like these different websites. So that's why it's a little bit when you go to look for papers, they're on different websites, but there's a reason for that. And so we have all these sort of websites with different sort of themes, and then we have within that theme we have things that need to be synthesized, and so that's an overview of representational brains and phenotypes.
Well, one of the things that I have been building up to doing for other things, and I would offer to do here, is like what I'm turning JoPro into, like the main JoPro website, is essentially modeled after the MIT Media Lab. And I can either show my screen or just talk about it. I don't really care.
You share your screen, I think.
But
I'd like to see
because I think what I might do is propose something here. Let me see if I can make this easier. So this is the Media Lab. It's actually a very interesting website. I've spent a lot of time diagnosing and dissecting it. So what's cool about this is that the Media Lab itself, the front page of it is just a bunch of these like updates and posts. If you look on the bottom left, I think you can, I don't know if it's going to show on the screen share, but you can see like the URL preview is like post or publication or like groups. And they have basically just put everything as one of these tabs here. Then you have the menu or like research about, right, whatever. But like let's do this one might work. So here's a post about something that's sort of like a news article. But then they have topics and people and projects and groups. Like this is the responsive environment groups. They have their cool little MIT logo, whatever. And in the group you see people and all this other stuff. But it's also, yeah, it's great. Here's a group website. And this will take you to like the equivalent of the representational brains and phenotypes page, right? And I'm realizing like what I might suggest doing is essentially, GeoPro is acting a lot like what the main, like what this page is, or like as a place to house stuff. It's not necessarily a, oh, this is kind of an awkward website, I suppose. Well, no, it's fine. Actually, it's a really cool website. Excuse me. It's a very cool modern website, and I didn't think it was. But essentially, the Media Lab stuff is sort of almost a landing page for many different labs and projects. Like if you go here, it's well spelled out in terms of like, this is a nice way they talk about it here. It loads. It is taking a very long time for this page to load. But like, yeah, blah blah blah. Dozens of research groups, initiatives, centers working on hundreds of projects. So they break it down through projects and initiatives and all this stuff. And I feel like what I can try to do, if it's of use, is make a little bit more of a front page for these different, I want to show the things in the lab, but the, like the Phasix project, or I don't know if that's really a project or not, but like the Phasix thing, the Phasix entity.
Yeah.
And essentially give it more like kind of a front page. Not that it, maybe more like landing page, because I really like the idea of having the separate, like this site is the actual page where the updates are, the news are, but having something essentially, I'll stop sharing my screen, but like essentially what I tried to do with that GitHub page a while ago, that was sort of an attempt at organizing things a bit more. I think it may be easier to do now in a different format.
Yeah.
So I don't know if what I'm saying makes a lot of sense, but like, I think there are ways to do that that might be help some of the organization, but then also funnel people to the right stuff in terms of seeing like Phasix work, stuff like that.
Yeah, well, I think, yeah, there is sort of a public relations aspect to the main Media Lab page. Like they say, okay, here are the new things that we're working on. Here are the ongoing projects. I mean, they have more press releases there, but also you can just basically, highlight things. Like we had the timeline, which was very hard to read or hard to get anything out of. It's just like, okay, at this time we did this. It would be nice to have, and it might be a little bit of work, of course, but to have like a, say this, why it's, we did this thing, why it's important.
Yes, yeah.
Have some resources connected, yeah.
I think what I'm working on with the GeoPro website right now is basically, I've made some custom. I've attempted to emulate some of how the Media Lab does. They have a nice way of breaking down people, projects, and the working groups. And I think that could be a nice way to separate out. Like if you go to the working group or a project, it will give a little description about it. And I think that's something I might try to work on, I guess.
Yeah.
And if I do make progress on it, I'll let you know. But it will. I think that would be a direction that I could see it being a good way to go, basically.
Yeah, I think so. That would be a good idea. Yeah, it would be, something that would take a bit of time to maintain, but would be good.
Yeah, and I know this is a really deep in the weeds and nobody cares about this, but me and maybe Bradley right now. But like, what I've realized is there's a much easier way than the way that I've at least done it in the past, where like if you funnel stuff in the right way, it won't require the maintenance and like all of the GitHub stuff that I've tried to do before were just kind of nice, but like very not evergreen enough. So I think there's some things that I found that I think are going to work well. So I'll try to like report on that over the next few weeks or so.
Right.
Particularly as we develop the other things and the internships and the model building and everything else too. So yeah.
Yeah, that would be great.
Step away from that.
Yeah. So that's great. Thank you, Jesse, for the update and for those all that great work in terms of laying out the sort of mentorship stuff and everything else. I see Trevor has joined us. Welcome, Trevor. Morgan, I don't know if you want to give an update before we move on to other things.
Thanks. Yes, sorry for being late today.
Oh, that's fine.
Spaced it. So.
I was just gonna say, sort of relating to Jesse's talking with Soni people, that I think the link that you posted about was it centering AI in medicine or AI something like that. Anyway, they were partnering with SUNY Downstate, and in terms of neurotech, there's two really interesting groups at SUNY Downstate. One is Merrim Bixum's group, which is focused on neuromodulation. So, kind of one of the lead groups in TDCS. So electrical stimulation, but really does follow lots of those. It organizes the Neuromotic group, or the Neuromotic conference that just happened, and is a huge draw. I believe at neuromotic.org, and so it's a really good group on that front. And then the other group, and I'm blanking on his name, except that I know that it's very Spanish. It's like Salvador somebody, and I'm blanking on it, but it's the NetPine, pyne.org, developer or lead, and so that'd be like a computational neuroscience group, and they work in addition to, it's kind of an extension of Neuron, the software package and project, which is a really bad name for Google searching, but together with sorry, I'm forgetting the Metacell. Metacell does their like GUI front end, and Metacell does that kind of software contract work for a lot of open source projects.
Right.
Yeah. Anyway, just wanted to mention those two. Just when I saw SUNY Downstate, I was just like, oh well, at least on that neurotech front, I don't know if that helps maybe kind of refine some project ideas or things like that. But definitely, like that, those would be two really great resources from SUNY Downstate that would be I would highly suggest to link up with if there's students interested. I think they referred to it as like links to SUNY Downstate for translational and clinical research.
Yeah. That's great. Yeah, thanks for that update. All right, so in one of the, I don't remember what channel it was, I think it was Morgan who posted this paper here, or it could have been something I ran across on social media as well. Anyways, this is a paper that came out this week on the arXiv.
It uses Doom, so we all saw it.
Yeah. So this is so this is a diffusion model via video games as world models. So this has world models in it, diffusion models, and which we will talk about more in the coming meetings, because I've been, our upcoming paper on the overhead regulator theorem talks about diffusion models a lot, and there's a lot of really good, interesting work on diffusion models coming out. Plus, I've been doing a deep dive on anti-diffusion models, and it'll be interesting to talk about more in the coming weeks. But anyways, this is an up-and-coming area of machine learning, and what they're doing here is they're using video games, basically, where they're looking at Doom and they're building these world models, which are these models, where you build representations of as much of the world as you can and provide it to the model. So that's what they're doing. They have a site, gamegen.github.io. That's, I guess, their open source repository for this. So this is a place where you can kind of actually get the code to implement this. In any case, so let's go through the abstract and read. This is, I think, from Google, especially DeepMind. And so let's go through the abstract and see what they're doing. So they are using Doom here. And these are screenshots from Doom. And they kind of have this world model of Doom, I guess. And they're able to get like these representations so the AI can look and navigate the space. So it's a nice way to like kind of encapsulate a world model because you have a finite world in the game. You have these different scenes, you have these different moves that you can make. And I think we talked about this with respect to like maybe some of the sequential models that we've talked about or some of the large language modeling that we've talked about where they're representing like spatial cognition as like this sequence of events that you do. So it very much relates to some of those papers as well. Okay, so the abstract reads, we present Game Engine, the first game engine powered entirely by a neural model that enables real-time interaction with a complex environment over long trajectories at high quality. So this is this Game Engine model. It's a neural model that enables real-time interaction. This complex environment is this game here that they're playing. They're showing different screenshots of this first-person shooter, and you're going through the game. It's, again, a human player playing Doom at 20 frames per second. This is what's being, the model's being trained on. And you have this, these long trajectories of action sequences. Game Engine can interactively simulate the classic game Doom at over 20 frames per second on a single TPU. So this is Game Engine being able to simulate gameplay in Doom like maybe like a human would play. And, it doesn't take maybe like a human would play and it doesn't lag and try to make decisions. It can just, the action can flow like a human player would play. Next frame prediction achieves a PSNR of 29.4. This is a metric that they talk about in the paper, comparable to lossy JPEG compression. So this is something that is not, out of the realm of like, it's not computationally expensive necessarily. Human raters are only slightly better than random chance at distinguishing short clips of the game from clips of the simulation. So this is where, you ask humans, can you distinguish the gameplay basically that the simulation is doing and the gameplay that the human is doing? If I were to show you clips of the games, would you know that a human was playing it or the AI? Basically, they do this rating. They find that it's just a little bit more likely that you would detect an AI sequence from a human played sequence. So that's pretty good. Game Engine is trained in two phases. The first is that an RL agent learns to play the game and the training sessions are recorded. So there's a reinforcement learning agent that plays the game, the training sessions are recorded, and then the human rater, judges whether that was an AI or whether that was like a human. You can also get this extract this world model, and examples of the RL agent playing. So that's the first stage of training, is that the RL agent learns to play this game, and the training sessions are recorded. Then a diffusion model is used independently of the RL agent to produce the next frame, and this is conditioned on the sequence of past frames and actions. So we take that recording, we basically condition the diffusion model to, give it information about that world, and then train it to produce the next frame. So there are two stages. There's an RL agent training stage, and then there's the diffusion model that's predicting the next stage. And the thing about diffusion models is that diffusion models are basically trying to recover images or recover things that it's been trained on and tries to predict what that looks like. So for example, a classic diffusion model example is where you have like a set of pictures and you might noise them with different types of Gaussian noise. And if you keep noising with Gaussian noise, the images become indistinguishable from the background noise. So what you want the algorithm to do is you want it to get like basically the plain images, and then train the model on progressive noising of these models, of these images. And so if the model has all that information, a diffusion model can basically predict how the noise sort of obscures the image, and then it can recover the original image. So a diffusion model is basically, working from sort of this noise as the mask, and then, it can recover an image from the mask. Another way to think about that is that the model hasn't seen certain things in the world, but it's seen other things. So it sees these images of the game, and then it can, given like discrete images that are separated in terms of action and gameplay, it can, given the full range of states of gameplay, it can recover what the next stage should be, because it's basically recovering that from noise, the noise being the inability to observe those intermediate states. So that's what they're using. They're using this ROL agent and this diffusion model. Okay, conditioning augmentations enable stable autoregressive generation over long trajectories. So they can generate these gameplay trajectories and using this type of approach. So they talk about computer games and how it's centered around the following game loop. You gather user inputs, so there's a user that does something. The game state is updated, so if you move left, the scene will rotate left, and you'll go down that path. So the user gives the input to rotate left, the game state shifts to the left, and then continues in that direction, and then all of that action has to be rendered to the screen. So these images are rendered by the game engine. But this all has to be seamless. It can't just have a lot of lag in it. It can't have a lot of noise. It has to be seamless for the experience to be good. This game loop, running at a high frame rate, creates the illusion of an interactive virtual world for the player. Such game loops are classically run on standard computers. And while there have been many amazing attempts at running games on bespoke hardware, such as Doom, being run on kitchen appliances such as a toaster and a microwave, a treadmill, a camera, an iPod. I guess the thing is that Doom is like, you can play this game on devices that have a really low amount of memory. I was watching a video this week actually on Doom. Someone trying to take Doom and make it into a non-Euclidean game or something like that. It's interesting stuff, but basically Doom is, there's this ability to take Doom and to run it on different types of devices. You can also play Doom within the game of Minecraft. So I'm not really sure, this is from a Reddit post here. It runs Doom. So there are a lot of things that run Doom that aren't the best hardware you can think of. It's just kind of like random hardware. Like my microwave can run Doom, my camera can run Doom, my iPod can run Doom. Things you wouldn't think that could run Doom. Okay. But in all these cases, the hardware is still emulating the manually written game software as is. So you can run Doom as is in a lot of different contexts. And so this is their point here. So what that means is it's easy to break down the game. It's easy to build a world model because there's basically a world being represented here. But it doesn't, it apparently doesn't require like a lot of computational overhead to simulate it. So that's it. Okay. So furthermore, while vastly different game engines exist, the game state updates and rendering logic in all are composed of sets of manual rules, programmed or configured by hand. Okay, so in recent years, generative models have made significant progress in producing images and videos conditioned in multimodal inputs, such as text or images. At the forefront of this wave, diffusion models become the de facto standard in media. So this is where, diffusion models are sort of a generative model that's sort of the new model du jour, basically. So it actually turns out that diffusion models, because they're based on physics, and a lot of the diffusion models in physics, and a lot of the things we're doing in like computational simulation, they actually make very diverse, versatile models. So we can use diffusion models in a number of ways. that actually enable a whole new set of this is why I talked about it in the Evergood regulator theorem paper, because it opens up a lot of avenues between, say, like machine learning and, say, physics or theoretical computer science. At a glance, simulating interactive worlds of video games may seem similar to video generation. So platforms like Sora, Stable Diffusion, DALL-E are all doing image generation. They're all doing these generative models of images and video. And so, we think about the way that these models work with video generation. They need to be very large to get like good videos and video generation. And so we might think that we need this for video games as well. But we actually don't need that, because as it turns out, we have we don't need as much computational power for video game generation or this interactive world simulation. So in that sense, it's a lot more compact than video generation. So interactive world simulation is more than just very fast video generation. The requirement to condition on a stream of input actions that is only available throughout the generation breaks some assumptions on existing diffusion model architectures. Notably, it requires generating frames autoregressively, which tends to be unstable and leads to sampling divergence. So this is where, we need actually different requirements from video generation. So to condition on a stream of input actions, we can only, it sort of breaks some assumptions of diffusion model architectures that currently exist. So a lot of the diffusion architectures that we use for Sora and Stable Diffusion and DALL-E can't really be used here because they do break some of these assumptions. So it requires generating frames autoregressively, which tends to be unstable. And this is another point in the Evergood regulator theorem analogy to diffusion models, is that you're, to make predictions in time series, which is what you're doing here, you need to have some sort of autoregressive model. In any case, I'm getting too far into the weeds of something we're not looking at directly, so I'm not going to spend time on that. But they do talk about world models here. Several important works, and they talk about Hans Schmidt-Huber from 2018, Kim from 2020, and Bruce from 2024, where they talk about generating world models from video games. So especially in Hans Schmidt-Huber, they're simulating an interactive car racing game in a video game environment. They're sort of training a model on that game, and then they're reproducing the game in different ways. So, a lot of this work on world models is focused on interactive video games. Nevertheless, most of these approaches are limited in respect to the complexity of the simulated games, simulation speed, stability over long time periods, and visual quality. So we might ask, can a neural model running in real time simulate a complex game at high quality? And so in this work, they demonstrate the answer is yes. So back here, I made a statement that seemed like it was contradictory, which is that, interactive video games and video generation are similar, but interactive world simulation requires something else, something more, apparently, because it's not just very fast video generation. You need this autoregressive component. And so down here, that may suggest that we need an even better model than the standard diffusion model. But another way to do this is to actually pick a game that's very compact. And so that's what they're doing with Doom. And so they show that a complex video game, the iconic game Doom, can be run on a neural network. They're using a version of the Stable Diffusion 1.4 model, which is from Rombach 2022. They're running this model in real time. They're achieving a visual quality comparable to that of the original game. While not an exact simulation, the neural model was able to perform complex game state updates, such as tallying health and ammo, attacking enemies, damaging objects, opening doors, and persist throughout the game state over long trajectories. So you can do all these things, these interactions with the game. The game can be rendered based on those decisions and things that are going on in the game, and then these can persist over long trajectories. So that's what they're doing with this game engine platform. And so Game Engine itself answers one of the important questions on the road towards a new paradigm for game engines, one where games are automatically generated. Similarly to images and video are generated by neural models. Key questions remain, such as how these neural game engines would be trained and how games would be effectively created in the first place, including how to best leverage human inputs. We are nevertheless extremely excited for the possibilities of this new paradigm. So this is kind of showing Doom in terms of what they're doing. So in a world model, this is basically a world model of the game. So what they're doing is they're using a methodology similar to Hans Schmidt-Huber. They're building this world model of the game, and they're recovering the different components of the game through this world model. The second one is this game GAN. So they're using a generative adversarial network to model the game, which is called Game GAN, and they're actually able to reconstruct the game a little bit better, but it's still blurry. If you look at the Hans Schmidt-Huber paper on world models, they recover components of the game, but it's blurry, and it's because it's kind of picking up on some of the features, but it's not doing it the way they're doing it here, which is on the right, and that's actually recovering the game fully. And of course, you pick a game that's that's amenable to this, but you also build the way use the methods that they're using here. So this is and they can show this clear improvement in Doom over the other methods. So they give their methods here. They have this generative model training. Basically, they have the frames and the actions. The frames are in blue, the actions are in yellow. This goes to previous actions. So the current actions go to the previous actions. They have this reinforcement learning agent that observes the game environment, observes things are rewarded in the game environment, and then takes a corresponding action. And then during the training of the RL agent, the episodes are stored as these frames and actions. The actions then go towards these, they're compared with the previous actions and the current action. So you have the previous actions, a store of those, and the current action A n-1, and then that leads us to build an action embedding. And this is now part of the generative model and training the generative model. This action embedding then tells basically the game all the possible actions that can be taken in the game. So you go through this RL agent, the filter of this RL agent. Throughout the training of the RL agent, you get these episodes that are stored. You get these frames and actions. The actions are stored into this action embedding, and then the frames themselves are encoded to latents, which allow you to predict the next frame from the current action and the available frames. So you have previous frames, the next frame, that's encoded to latents. That then goes into this denoising network. And the denoising network, remember, it's this diffusion models, so you're recovering a signal from noise. You're pulling the signal out of a bunch of noise, the noise being everything in the action embedding and the latents, and you're pulling out the current frame that's predicted with some diffusion loss that goes back to the latents and is kind of improved upon as the model is trained. So that's how Game Engine works. It uses the reinforcement learning agent, the diffusion model, and it puts those together in a way that improves upon the world model and a generative adversarial network, and even like a standard diffusion model. Okay, so that's all I think for that paper. There's a lot more technical detail I'm not going to get into, but I think that's a good kind of overview of things. And so I see Trevor has his hand raised and Morgan has his hand raised. So let's.
I was pointing at Trevor.
Okay. Yeah.
Oh, I appreciate it. I was just curious. The actions were the actions predefined in that paper, or did they like do discovery and like? I'm curious how combinations of actions were happened and all that.
Well, in an interactive video game, the actions are just what the agent will take. So, like, if a human plays a game, the human will shoot at a target, or they'll be rewarded with points if they pick the right target. So those things can be learned by the RL agent, or in the human case, it can be learned. But there's this sort of directed towards things that make sense. Like you maximize your reward in the game environment. You navigate towards different parts of the game world. And all those things are stored in the action latents. So the actions are actually generated by playing the game itself. It could be the RL agent playing the game, it could be human playing the game. And those seem to match well. That's what the paper said. And so that's where you're learning your actions. They're not just generated at random. Although it'd be interesting if they did do a randomization of it to see what that would look like.
Interesting. All right, so I'm assuming there's still some like base level actions, like kind of at the beginning of the paper, you're just like go left, go right type of thing,
Right.
And then it just builds up into this latent thing. Okay, interesting.
Yeah. Also the thing is that you're building upon your previous actions. So if you go left, you're going to have a certain part of the world that you're exploring. If you go right, there's another part of the world you're exploring. If you play it multiple times, you can explore the world more and more, so you get a better handle of the world. If you've ever played a video game, where you have to play like, I don't know, for 3 or 4 months to really learn it. That's what's going on, is you're learning these different strategies by exploring the game world, exploring different things that you do, and then kind of getting a sense in your head of how to play. And then eventually you win because you get really good because you know that environment intimately, and you can navigate it pretty easily, and then choose the actions that are the best in that situation.
Yeah, it makes sense. I guess one of the things that first comes to mind is a few years ago at some NIPS contest, there's like a text-based game. I'd have to look it up. I can't remember. Meta really loves this thing, and like the winning solution was like a hand-coded hierarchical strategy. And I'd just be curious how a world model like this would hold up to something like that, because it seems like there's this interplay between this world model being dependent upon how it was trained, and so just curiosity.
Yeah. Well, there's this whole problem of what world models are, and so the whole idea of a world model is that you can train a model, but it can only do things on what it sees. So like if it's seen something before, it can like classify it. If it hasn't seen something before, it either has to generalize from the existing data, or you're out of luck. It doesn't, it misclassifies things. And so people come up with the world model as a way to sort of say, okay, we can just build a representation of the world. So instead of training it on like letters and numbers, we can do that. But we can also provide the context of the entire language. And that'd be a world model of a language or something like that. And then the game would be like, I can just simply train it on playing the game, which would be, just it would know what it sees until you'd have to provide it with every possible scenario again and again to get, but you can also just build a world model which doesn't require explicit game sequences, but just requires like every scene in the game to be there. And, so you have this model of the world. And so if you train the model, it has that information, but it can put it in a context, which is the entire world. And so that's what people, and this is an area that's not, been fully explored. It's kind of like, people have introduced papers on it, but it's one of these things that people haven't really refined as much as like some of the training, like training an RL model. We know how to do that. A lot of people have written a lot of papers on this, but world models aren't that well studied. Like we had a handful of papers on them, so relatively speaking. So it's an interesting approach, and it's definitely one that overcomes that problem of generalization. In other words, if I generalize, I am taking the model's basically taking a leap of faith in the sense that it doesn't know if it's the right answer. It's just kind of saying this is similar to this thing, so that's the answer, and it may be right and it may be wrong. The error rate may be very high, so that's bad. A world model, at least the model can compare it to something in the world model. Oh yeah, that's a feature that should be in the world, and so it would be, better suited for it.
Yeah, just this is somewhat just my armchair ignorance here, but it seems like for something like this, if you're trying to like model the real world, you would want some sort of easy way to exchange between the world model and the simulator. And
Right.
so, like whether it was encoding objects that you could then throw in a simulator, because there seems to be like this dependence on simulation to teach the world model, and so yeah, I don't know much about this world, but I don't know if there are approaches that enable that type of thing.
Yeah. So let me go to the paper. I want to show something here. So they talked about world models up here somewhere. Oh yeah, Han Schmidt-Huber 2018, Kim 2020, and Bruce 24. So if we go to those citations,
The Han is obviously the first one.
yeah. So Bruce is, let's see if they have it here. How do they have this organized? So this is Han Schmidt-Huber world
You're
models.
not sharing your screen, just
Okay.
so you know.
Yeah.
Oh, thank you.
All right, let me go share my screen again. All right, so the first one is Han Schmidt-Huber. This is world models. This is arXiv paper, even though they don't mention it here. But that's the one, I think the first one that was based on the racing video game. And then I said, what, Kim and what was the other one?
Oh, I forgot here in December.
So this is the Kim paper, I believe. This is learning to simulate dynamic environments with GameGAN. So this is the GameGAN paper where they use a generative adversarial network. This is at the IEEE conference on computer vision and pattern recognition, CVPR. So what was the other one? Did you remember?
Did you say Bruce?
Bruce, yeah. Let's see if I can find that. Should be maybe up further. I don't know how they have it organized. I think it's, here it is, Jake Bruce,
There, yeah.
Michael Dennis, Ashley Edwards.
Jeannie.
Yes, Jeff Kloon is on this. Nando Diffusion.
Yeah, see, like a whole bunch of people.
So Genie, Generative Interactive Environment. So this is from 2024. This is also an arXiv paper. And, again, they're using these generative models. There's this difference between like maybe video generation and interactive environments. Video generation just needs to like put together a sequence of things. The interactive environment needs to take inputs from the player and put it, use it to guide these worlds. So the player determines how the world is explored, what the sequence should look like. And that's an autoregressive process. And yeah, so there are a lot of interesting statistical properties here, that differ between the two. And yeah, they have a lot of interesting stuff in this paper. They do this autoregressive evaluation and context length, noise augmentation. So it's pretty cool. Yeah. Any other comments or questions about this paper?
Well, it's as usual. I'm just upset I didn't do more background on it before, because it would be good to, one, put it, like you said, put it in context of the, not necessarily Han Schmidt-Huber, but the specifically some of the world model papers that are in the RFP.
Oh yeah.
That like Saffron's RFP,
Yeah.
right? Like some of his links are really interesting because again, they're all kind of focused, or certainly like the progression of papers is focused on kind of having more and more foresight. I mean, like being able to kind of, have a better memory and kind of make better plans in the future.
Right.
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 And so you see at least again in some of the staff room papers, but like specifically some of those references. Sorry, Trevor, I don't have them off the tip of my tongue, but yeah, just because again, like so much of this, it kind of like it's subtle. It only comes out from the specific evaluations that you're making, or like the metrics you're tracking and things like that. Like at least for me to get a intuitive idea of what these design choices are getting you.
Yeah.
Anyway, that's
yeah.
Again, that's more just there's a long way of me saying like I wish I'd done more background prep.
I think the use of Doom is really interesting here because it's like Doom is like something you can program onto some like very trivial piece of hardware, and that means that it's something that has a structure that's unique to this problem, I guess, or can show this problem very easily. It's kind of like the elegans of like computational models that you can use.
Well, I mean, certainly the video, video generation versus video generation as world models.
Yeah.
Like that was an interesting thing. Remember when I can't even remember. It tells you how much hype there is in this area. I can't even remember the 3D video generation tool, like from OpenAI or somebody. Like remember that you can make
Sora?
these.
Oh, Sora, yeah, with Will Smith eating spaghetti. Yeah.
Yeah, and it's like whether that meant that world models were solved or something like that,
or
wow. Like that you had world models, like again, like it's not
yeah, they didn't really solve it. Yeah.
there's definitely more, as well as it just being the vocabulary more specific, in terms of what RL people are going for with these world models,
Yeah.
And especially with it's kind of, and I wasn't sure if Trevor was thinking about this, but again, like stringing actions together is the kind of thing where you start to look for like tool use, right? Like tool use require memory and like being able kind of concepts or representations, potentially.
Yeah, I think there's something about these sequential representations, because it seems like that's what people are kind of really interested in right now, and how those can be handled and how you can extract those and learn. And there are a lot of statistical properties of sequences that are different than like just kind of putting in independently distributed data. And I don't know what the connection is there, but yeah, something worth thinking
about. Yeah, LeCun seems to really be emphasizing this. He talks about task planning and all that. I guess that's what a lot of the labs are focusing on these days.
Yeah. All right.
I mean, the ones that aren't focused on the language models.
Yeah. All right, that sounds good. Well, let's call it a day. Thanks for attending. And Trevor, yeah, you might find Lukas's talk that he gave earlier in the meeting interesting. You'll see it on the recording. It was on lipid metabolism, genes and proteins in C. elegans, and they were doing a lot of interesting work with that. So
check out the new build a cell seminar.
Oh yeah, okay, they have a new one.
Yeah, it's a good cytoplasm, and it's somebody from Ski at University of Utah.
No kidding. Huh.
Yeah.
I wonder who it is. I'll have to see if I know him.
Yeah, it's good stuff.
Yeah. All right.
Yeah.
Thanks.
Thanks so much.
See
Have a
you
good
later.
one.
Take care.
Take care. Bye.